In today's lecture, we're going to talk specifically about bacteria. So bacteria are all prokaryotes, meaning they have cells that do not have a nucleus or any complex organelles. And if you recall, they were actually split into two separate domains, archaea and bacteria. So the domain bacteria contains the kingdom Eubacteria. And Eubacteria, here are their main characteristics. They have cell walls made out of something called peptidoglycan. Archaebacteria, which belong to the domain archaea, have cell walls made of a different material, isoprenyl ether. They have different cell membranes, but here's the only thing you really need to know as far as test goes. Archaebacteria versus Eubacteria. Think of Archaebacteria as ancient bacteria, the first ones that ever lived on Earth, and we find them especially in harsh environments, like methanogens, which live in swamps, halophiles, which live in salty places, thermophiles, which live in really hot places. So archaebacteria are the ones that are sometimes called extremophiles. They're found in extreme environments. And then Eubacteria are going to be the ones that we find sort of in our everyday lives. They do good things and bad things. A lot of people are familiar with E. coli, or lactobacillus in yogurt, or strep that causes strep throat. So those are all Eubacteria. And here's some pictures. You'll see they look almost the same. They do have differences between them, but they're very tiny. So in our class, we're not really going to get into the details of those differences. It's something you would talk more about in, say, a microbiology class in college. Okay, so let's focus on Eubacteria. So Eubacteria tend to be classified by, number one, their shape. Number two, their cell structure, like what their cell walls are made of, how they move, and how they metabolize, meaning how they use energy. So the three main shapes, there are some others, but the three main ones are cocci, which are round bacteria, spirilla, which are corkscrew or think of spiral bacteria, and then bacilli are rod-shaped bacteria. And here's a diagram that shows all of those. So cocci, or the single is coccus for that. These are the round ones. Interestingly, like streptococci, which cause strep throat, strep actually means strains, so they're found in strains. And staphylococcus, which causes staph infections, staphylo refers to being in clusters or grape-like clusters. So even though both of them are cocci-shaped, round, they're found under a microscope. The way they sort of hook to each other is different. This is bacilli, so they are rod-shaped. And then really just know this one, spirilla. Again, there are other shapes in between, but this is the spiral shape that we were talking about. So spirilla is that shape. As far as their general parts, all bacteria have cell walls. Now they can differ. There's what's called gram-positive and gram-negative bacteria based on whether they have this thicker cell wall or a thinner cell wall. And different antibiotics work differently or work better against different types of bacterial cell walls. They all have a cell membrane, they all have DNA, they are living things, and they all have ribosomes, which make their proteins. And sometimes they can have a flagellum or pili. Now flagella would let them move if they have them, so they look like this. And pili, these are actually pretty cool. They're almost like little tubes, and they would let one bacteria hook to another bacteria, and they can pass little pieces of DNA called plasmids between each other. This is how one bacterium could turn another bacterium into an antibiotic-resistant bacteria, for example. So these are the main parts they all have. Again, this cell wall can differ in thickness and its structure, and that would actually be one of the things they might test is which kind of cell wall a bacteria has to determine what kind of antibiotic to give you. General characteristics, so some of them can move, again, if they have flagella. They can also have a slime-like layer that protects them. Some of them are autotrophs, so there are bacteria called blue-green bacteria that can make their own food. And then most of them, though, are heterotrophs, so they have to eat their food. There are three kinds of general metabolism that we might see based on what their oxygen requirements. So obligate aerobes are obligated to be in the oxygen, so they have to be in air. Obligate anaerobes have to be without oxygen. So for example, I know botulism is an example of a bacteria that's an anaerobe. I believe the Clostridium that causes tetanus and the one that causes gangrene, those are also anaerobes, that oxygen actually harms them. One of the treatments is to put your infected area in a hyperbaric chamber and flood it with lots of oxygen, and that actually damages the bacteria. And then there's what's called facultative anaerobes, which basically means they typically live in oxygen, but they can live without it too. They can just do fermentation or whatever, and they don't have to have oxygen. So obligate aerobes have to be in the air, think of it that way, have to have oxygen. Obligate anaerobes, surprisingly, are actually hurt by oxygen. They have to be in areas without oxygen. And then facultative can kind of go either way, with or without. As far as their growth and reproduction, they divide by something called binary fission. Very quick, faster than mitosis, and they make exact copies of themselves. They do not do sexual reproduction. They only divide by binary fission and make exact copies. The other thing they can do is something called a conjugation. And again, this is where two bacteria would hook together with one of these pili, and they can pass DNA back and forth. And this is a way that they could actually gain, say, antibiotic resistance by one of them passing a little piece of DNA that codes for the antibiotic resistance, passing a copy of that to the other bacteria. But that doesn't actually make babies, so it's not really sexual reproduction. They don't make baby bacteria from this, but they do exchange information. Some bacteria also can make something called endospores when conditions are bad, and this would allow them to stay dormant for months or even years. For example, I know botulism bacteria can end up in canned foods, and they'll form an endospore, and they'll just sort of stay there until conditions are right, and you can get a really bad food poisoning from those bacteria that they actually cause a disease called botulism food poisoning, which is extremely serious. That Botox toxin we use for all kinds of cosmetic purposes today, but it's really a very strong poison. It paralyzes your nerves. This is just a picture of binary fission. They basically copy their DNA and they split in two. Very, very simple, no spindle fibers, much simpler than mitosis. And this is showing conjugation where, again, they use a pili and they send a piece of DNA. Again, technically, this doesn't make babies, but it does allow them to change their genetic structure. Okay, just some good and bad in the bacteria. So how are bacteria bad? Well, the way that they cause infections in us is by literally either eating our cells or by producing toxins that kill our cells. A lot of times when you get food poisoning, it's not that the bacteria are directly eating your cells for food, it's that their waste product is a toxin, and that toxin is making you sick. But unlike a virus, remember, viruses hurt you because they basically take over your cell's machinery and your cells just make more viruses. In this case, the bacteria are alive. They're using your cells for their metabolism. So some pictures. This is strep throat, so these are your tonsils, and this is all bacteria growing on the tonsils, which is pretty nasty. This is cavities. Cavities are caused by bacteria. They're not eating your cells, they're eating the sugar in your mouth, and their waste product is an acid. And the acid basically deteriorates your teeth, and that's what causes the cavities. Food poisoning, again, is caused there's E. coli food poisoning, there's botulism food poisoning. Tuberculosis is another example. This is actually on the right, flesh-eating bacteria. It's a staph infection, very scary. It actually eats the layer between your inner layer of skin and your outer layer, and so the outer layer can't get any, there's no blood vessels there. So the outer layer of skin dies, and it just rots. And the only way they can treat it is they have to literally cut out all the infected area and do skin grafts. So very, very scary, that flesh-eating bacteria. So these are some things that bacteria can do. Now, there are, I should say, a lot of important jobs of bacteria too, though. Number one, they are decomposers. So one of the main things, fungi also do this, but one of the main things that bacteria do that's good is they break down dead stuff and recycle all those nutrients back into the soil so that it can be reused. So if stuff didn't rot and break down, all the nutrients in that dead organism would be trapped forever. So decomposers are extremely important. They also do something called nitrogen fixation. They basically take nitrogen out of the air and they turn it into a form that plants can use. Every living thing needs nitrogen to survive. But even though our air is 70% nitrogen, we can't do anything with that. We inhale it and we exhale it right back out. So how do we get our nitrogen to make DNA, to make proteins, all these important things we need nitrogen for? We actually get it from eating plants, and the plants get it from these bacteria who take it out of the atmosphere and turn it into a form plants can use. Specifically, like in soybeans, there's a particular bacterium that lives in their roots and makes nitrogen, a particular type of nitrogen-rich substance that the soybeans can absorb, and then we can get nitrogen from that. E. coli in your intestines, we think of E. coli as bad, and they are if you eat them, but they actually live in your intestines naturally, and they make, I believe, vitamin K. So sometimes it's important to have bacteria on your skin to protect you, in your digestive tract to make vitamins for you, and then, of course, around us in the soil as decomposers and nitrogen fixers. We also use bacteria to make certain foods, like yogurt is made using lactobacillus. We make other things. Some cheeses are made using bacteria. Wine is made using bacteria's fermentation. So they're foods and drinks and things that we actually make using bacteria. We also can use bacteria for genetic engineering to make some medicines and things. So there's all kinds of benefits. We don't think of bacteria as being only bad. There's good bacteria and there's bad bacteria. All right, if you do get sick with a bacterium, there are treatments. So before you come in contact, you can get vaccines against some bacterial infections. Like tetanus is one. If you've ever had a tetanus shot, that's against a particular type of bacteria. Diphtheria is also another type of bacteria you can be vaccinated against. So vaccines are good for certain bacteria, as well as viruses we've talked about. But antibiotics are only for bacteria, not viruses. So antibiotics like penicillin, streptomycin, ampicillin, they can kill bacteria, and sometimes they'll help your immune system fight off a bacteria. But there are strains of bacteria that are resistant. MRSA is an example, methicillin-resistant Staph aureus, I believe. So if you get sick with this bacteria, almost no antibiotics work against it, which is really dangerous. And the more we overprescribe antibiotics, the more we kill off the ones that aren't resistant, and those resistant ones take over. Disinfectants, you can't take a disinfectant, you can't drink Clorox, but you can use disinfectants on surfaces. You can sterilize things with heat. Milk, we pasteurize. We can store food in the refrigerator.